everybody. I think we'll go ahead and get started. Give just a couple minutes for folks that are coming in from the cold. But I'm happy to introduce Joe Dauer. Joe is coming to us from the University of Nebraska-Lincoln, and I'm really excited about the things that he's going to talk about today. So I really appreciate everyone coming and sharing with us today for his chat. Joe actually has an MSU connection. Many of you already know that, but he was here from 2010 until 2013 as a postdoc in my lab. So I really appreciate all that Joe contributed to our thinking at that time. I consider myself really completely new to the whole experience, and when Joe asked about working in our lab, I said there was one condition, and the condition was going to be I'm not going to be your supervisor. We are going to be collaborators and figure out what we want to do in this field of model-based reasoning and model-based learning. So I really appreciate all of the investment that he made in that effort. Joe has an undergrad degree from, you just told me?
Western Washington.
Thank you, Western Washington, and then went to Penn State for his PhD, and then to MSU. So he has a track record and a history of being in ecology and ecological science and weed science in particular. He made that transition to discipline-based education research during his postdoc time here. So he's really developed that quite a bit since then. I know that he and his wife together, you may know his wife or know of his wife, Jenny Dauer. She was a postdoc with Andy Anderson for several years while they were here together. And Joe and Jenny together have done a lot of really innovative work in the curriculum at UNL. So with that, you're going to tell us about
Cognition and neurobiology.
All right.
Yes.
All right. Thank you so much.
Well, yeah, thank you for all coming, and thank you so much for the invitation from Create for STEM and to come back. It's great to be back. So I haven't been here in 6.5 years or so into Lansing, but it is great to be back. And so the work I'm going to talk about today really did come out of this work with Tammy on trying to understand students' cognition during these modeling tasks. So I'll talk about some of the modeling we're doing, the work we're doing today down at Nebraska. And then, so these, I think it's helpful to really attribute that this is not just something that I'm working on in isolation, that there's a lot of people, a lot of collaborators. So people, some of the postdocs that I've been working with, oh, that's cool. Okay, all right. So some of the postdocs that have worked with me in my lab. So Tom Helikar is a collaborator on this computational biology. He's a computational biologist and does all the software development. And then the work that I'm going to talk about in terms of neurobiology is all being done with Carrie Clark. So Tammy and I and Carrie submitted a proposal recently to really look at, when I talk about the neurobiology, so we're trying to go beyond that, and it's all because of the work with Carrie Clark. So I am not a neurobiologist by training, and I've had to learn that field, and it is challenging, but really fruitful and interesting. And I hope you'll find it interesting also. So my work is really very focused on how do we get students who we would think about undergraduate students as really kind of on this, some of them on this more novice side of things, of understanding of the biological systems. They're able to organize components. They tend to relate them in kind of a linear fashion from A to B. And then, so we tend to think about them as having these models of a phenomena. So they can create a model and it tells us what's happening. But experts, we know, tend to be able to evaluate that model for quality. They look at very complex models. They really think about models for having a purpose, that purpose tells them about how that system works, how the components interact with each other. And so they really have models for a purpose. And so for me, I'm really interested in how model-based reasoning develops over time. And in particular, today I'm going to be talking about both the behavioral, so how do they change in their behaviors or what we see, so what they create and do, And then how do they change in terms of their cognition? So how they're processing the models, the biology, all of that, as they go from this novice towards expert. Now, I'm not going to suggest that we're going to get to that point where we're really understanding the expert side of it. We do understand what experts are doing, but I'm trying to understand this process that's going on. So there is definitely a process. So I'm going to give you just some general conclusions for the talk before we get going. This will kind of give you some cornerstones that this is what we're going to go to. I'm going to talk about 3 different studies, 3 different, well, the groupings of ideas. The first one is around conceptual models. So conceptual models really help students organize information. It helps clarify their connections of knowledge. So I'll talk about that to kind of get us all on the same page. So the foundational information. Then I'll talk about this study, this neurobiology study that I've been doing, and comparing students who are simulating versus reading about biological systems and talking about the neurobiology that's different between those. And so they really, even though they perform similarly behaviorally, there's very different things that are being done in the brain for those 2 different groups of people. And then the last one, I'll be talking about how students build computational models. So we actually challenge them to build a computational model that they have to then validate. And so we're going to look at what that means cognitively in terms of what we've found in terms of how difficult that is and what that tells us about their abilities to actually move beyond simulating, but also building those models, which is more like what an expert would be asked to do. So the reason that I've been interested in modeling is because modeling is a practice that scientists do. It is something that is just fundamental to science. So as Winshittle would talk about, it's developed these defensible explanations of the way the natural world works. And so modeling is just a part and parcel of being a scientist. But they use it in really different ways. So scientists are using modeling to explore these complex systems, to sort of understand what's unknown. So if you think about like a biochemical pathway, they're trying to figure out what are the pieces in between that aren't working, or what do I need to do in order to get X, Y, or Z to happen. They lead to this development of conceptual frameworks about understanding about how, like cellular respiration, if you could go back that far to like, how did all those pieces go together to understand how cellular respiration works, to make predictions. So if you think about things like the climate models, they're trying to make predictions about what's going to happen down the road, and also to generate these causal explanations. So why did this phenomena take place, and what do we need to know about all the interactions to get there? So they're using them in really different ways, but students don't really know that there are all these different ways. They just see it as something a task. Like, we need to go out and do this modeling. I don't think they really get to this understanding. And I'm not going to solve that today. But that's just to say that it's important that students become aware, but that we're also trying to get them to do more of these different types of things. So my goal in my position is really kind of twofold. There's the part that's really interested in the research. And so that's trying to understand how students change in their content and modeling knowledge. And then there's the part that's how does that inform the pedagogy? So how can we really go back and change? So the first one's kind of more research-oriented. The second one is more like the teaching-oriented. And so in all the conclusions today, I'm going to talk about in different aspects, like how is it related to researchers and how is it related to instructors? Because I think it's really important to be situating myself at that kind of interface between the research and the pedagogy, that it's not just me doing research for the sake of doing research, which is fun, and it's not just me teaching, which I do, and I enjoy that as well, but that there is an interface between those. And I would challenge you, if you have questions, I mean, that it is always appropriate to say, well, what does that mean for me as a teacher? Because I think that's really relevant for a lot of us who are teaching. Like, what's the point of this for a teacher that has to stand there and if you get up in front of a class, you have to say something. Well, you don't have to, but I mean, it's good to say something. And so, like, it's important to say something that's important for the students as well. So I need to talk about, this is the software that we use, and I'll be coming back to this. Well, so If you want more information about the Cell Collective, so this, the Cell Collective is the software that Tom has developed. They used the Cell Collective for computational biology to understand systems. He's a modeler. But this is the Learn.Cell Collective is the educational side where we have students interact with these interface where there's these models. Is there a way to make it just do the pointer? But that's okay. But there's a model of the interactions that students, and so we'll talk about, sometimes they're simulating, they're already constructed. Often there are parts to the lessons where they have to add or subtract nodes or change nodes. There's the inputs, which you would see in sort of a typical simulation kind of software. Then there's the lesson itself. And so as they go through the lesson, they change pages. And then there's the output about when you have certain conditions, what's going on. So this software is all, it's, as Danny Caballero was asking me this morning about, it's not an agent-based model. It's not calculus-based model. We're using Boolean logic. There are ways that you can go in and actually manipulate all the mathematical relationships. We don't ask students to do that at the intro level. But then we've had a lot of discussions about what is appropriate for introductory biology students to be doing with the mathematics. And that is a very open question, and I would be happy to discuss that. But we have tried to not make it totally black box, but not make it totally where you have to understand functional relationships in order to actually make it work. So we tried to strike that balance between those. But this is the software. And there's between 12 and 15 lessons. It depends on how you count them. Some of them for intro bio, some of them for advanced biochemistry, which is kind of an artifact of where the grant money was at the time. So the other part of this is I need to talk about cognition and the brain and how you process information. And the way that we accept that this tends to happen is there's this input. So you have some visual input that's going into this sensory memory. It's being processed into working memory. Some of that working memory is then encoded into the long-term memory, and then you retrieve that when you need it. So how many people remember what you had for dinner last night? Okay, most people. What about the night before that? So Monday night. Okay, a little bit. What about last Wednesday? Who remembers what they had for dinner last Wednesday? Okay, who said that? And so why did you remember that you had Chinese?
Because they came back from China.
Okay. Yeah, so here's the thing that's really important is that piece of information was encoded along with the fact that what you ate that day, right? So most of us, so if I asked it in a particular way, so maybe if I asked, like, who did you have dinner with last Wednesday, you might remember something about who you had dinner with. And then that might jog your memory about, now I have that piece of information. And usually when I eat with my sister, we have pasta, right? So, okay, now I remember what's going on. And so I think that the way it's encoded is really critical, and it really ties into how we retrieve that knowledge. And so when we think about our students, the way they stored that piece of knowledge is really important to how we ask that question. And so we have to be very understanding that when you write an assessment question and you say, I want you to create a model, or I want you to answer this true-false question, how did they store that model, or that piece of information, relative to the way you just asked that question? Because if you ask it in the wrong way, they're not getting it. They're not making that connection. They're not able to retrieve from long-term memory to answer that question. And so I use this analogy that I stole this picture from the internet somewhere about how we would store knowledge, store and retrieve knowledge. And so this house is sitting here on this cul-de-sac, and this go from the school to this cul-de-sac, you have to go a very particular way. You can't go any which way to go there. But if you store a piece of knowledge in this kind of cul-de-sacs around your brain, which is not a great example, by the way, if you know about neurobiology, but if you store it somewhere in a really remote location with very little connections, then you have to retrieve it in the exact same way. But if you have a lot of different ways that you can store that, so you could follow this red path, or you might access it through some different questions in a different way, if you can store it with a lot of connections, you can retrieve it with a lot of connections, or you can get there. So you might not remember exactly, but you kind of work through, oh, my sister threw the pasta. Oh, yes, we had macaroni and cheese because we had kids over that day too. So I think that it's important to remember that if they're storing knowledge in disparate places that are disconnected, it's going to be very difficult for them to pull that information out. And so I've used this because this is kind of the way I think about modeling, is that models help students organize information and so that they can retrieve it in a more efficient manner when they need to use it. So they can go from that long-term memory into the working memory along with new information, and they can actually answer the questions. Okay, so, so part of that is, so I'm going to talk about this kind of conceptual framework idea that, so the mental model is what's working in the student's head. It's what's in their working memory. It's that, how the information is organized. And for students, when we ask them to create a model of some sort, draw it or to write it on the computer, we're asking them to externalize what's currently in their head. And so that's this visualization or verbalization of that mental model in their head. And so it's something that we can have them do, again, on paper or whiteboard, whatever you want to do, something that's, it's a visual representation of what's in their head. And when they've done that, once you've put that down on paper, that allows students to revise the connections that are in their mental model. So you revise things based on what you're observing. So those are more strongly connected, or maybe I don't understand how those things go together, so I'm going to go gather some more information and think about it. It really changes how you think about the components, the relationships within that model. How are things going together? So this is I'm going to come back to this a little bit later about how the interaction between mental models, conceptual models, and then I'll layer on the computational models in a little bit. So those mental models, we can externalize those through these conceptual models, and we think about those as kind of this level one. So that allows us to show, or allow students to kind of show us what they know about their current mental model. But we're going to talk about simulating systems, so this comparison of your mental model. Now you've got a visualization of that data. Remember that output graph that was going along. So how does that influence your mental model? And then what is the cognitive change when you actually move to building that computational model? So when you need to actually go from just looking at a scratch piece of paper to putting something on a computer that's going to produce an output, and then what does that tell you about the system? So like layering that back onto the whole analysis and that revision of that mental model over time based around these mathematical representations in your model. So we do think of this as a hierarchy, that to be able to build a computational model, you do need to have a good understanding of building a conceptual model and having an understanding of what happens when you're simulating that system. And so from the teacher's perspective, we think about this as an opportunity to elicit those understanding of their relationships and maybe the function of that system. What does it look like? It allows the students to describe kind of thought experiments with that. The simulating systems provide this opportunity to really emphasize dynamics, like what happens to that system when I perturb it in a little bit, some ways. But you don't have to understand exactly the mathematics that underlie that in order to simulate the system. And then building the computational models really provides this opportunity to re-emphasize those dynamics, but also that data-driven revision of your mental model. So now that I see that it's not working, it's broken, or how do I know it's broken or it's not working? And then how does that change the mental model, and then the conceptual model, and then the simulation that we're doing. So they each provide different opportunities, and so we're going to talk about each of them in these 3 different studies about how those opportunities, what those can mean for an instructor. So the first one I'm going to talk about is what do these conceptual models tell us about students' biological knowledge? So in this case, we again are working with a cellular respiration context here. We are trying to get students to understand that the glucose contains these, allows this chemical energy to be used for work by cells. And so we're really trying to get students to understand inputs and outputs of these different systems. So glycolysis, or these different processes, glycolysis, fermentation, pyruvate processing, citric acid cycle, and the electron transport chain. And so they walk through these lessons in which they perturb it and turn it on and off, inputs, outputs, things like that. And students at the end were asked to create a computational, or I'm sorry, just a conceptual model of cellular respiration. So we gave them 20 possible components to include in their model. We asked them to link those with arrows, and they're supposed to write what the relationship is between those. Again, this is not anything mathematical. At this point, we just are asking them to relate those. Sometimes they relate those. Sometimes there are blanks you'll notice around. And then you also, some of these, we've gone in and scored those. So we actually score the quality of those relationships between a 1, which would be inaccurate. We called it 2 was vague. Like, we're not really positive that they understand, but it's not wrong, but it may be correct. And then 3 would be something we would say would be a technically correct relationship between those. And we also look at the organization, how many components did they include, and then how interconnected is it, and just to see, those are some of the broad metrics that we're using. And we see that students have a pretty good understanding of glycolysis and the citric acid cycle. So they include those, most students include those. There were 180 students, so you can see most of the students include those two. Fewer students, just about half of them include fermentation, so half the students are not even including the fermentation idea in their model, and around the same for electron transport chain. That's probably a reflection, like the fermentation is interesting because that's not really heavily covered. It's in there and it's involved, but the ETC and the fermentation kind of are, in the lesson they're not afterthoughts, but I think for students they become afterthoughts. Like, we were really interested up front, we've heard about these terms, many of them have probably heard about them in so these are tend to be freshmen, sophomores. I don't know, I'm sorry I didn't mention that. This is introductory biology students. That's all this whole thing is about with introductory biology students. So they're going to be freshmen, sophomore students. So they may have heard about this in high school, and probably some about this, but not a lot. But they don't tend to be included a lot, and then they tend to be not well connected. So we tend to see these being on the electron transport chain, maybe a little less so, but fermentation as kind of a peripheral idea that's kind of attached. And then actually we noticed that the percent of those that are actually even correct, so we rate it as a 2 and a 3, is about half of those are correct for those. So they tend to do a better job with glycolysis and citric acid cycle. In general, those electron transport chain and fermentation, they're just less familiar with it. They're less integrated into their models, and so they have lower quality relationship with these processes. So this section really, that these conceptual models are important for showing us that students what they're familiar with. This was done as a post-analysis of their knowledge, so they had gone through the whole lesson. For an instructor, it basically shows you where there might be gaps in the knowledge. If I was an instructor, I read this as, maybe we need to invest more time in talking about the electron transport chain and fermentation to make sure that they're understanding how it's integrated, because it's an integral part of cellular respiration. It's just not rising to that level for many of the students. And then the modeling here helps promote this visualizing of the connections. So students, if you ask them what electron transport chain is, they can probably spout off some version of a definition. The same with glycolysis and citric acid cycle. But when you try to put them into a model, it's hard. I mean, we know that it's purposefully hard. But it shows us that level and quality of that integration of those in such a way that maybe we should think about, so it's not just knowing what fermentation is. So as an instructor, just telling them more about what fermentation is, does not help them integrate it into their model. Like, we need them to be actively focused on which of those relationships don't they have a good understanding of, and like, how can we improve those? How can we get them to integrate it rather than just add it? Okay, so I'm going to move on to the second one. Are there any questions about the first one? Yes?
So, go back to the
This one or the data?
yeah, the data.
Sure.
Can you tell us something a little bit about how the assessment for the class itself, what was the actual assessment? Because one can argue that one way to validate this data is to compare quality of conceptual model to actual results on the whatever formal assessment is giving at the end of the class. And maybe that could be also like if a student scores high on this and very low on the assessment, that actually might be a bad assessment.
Right. Yes. No, it's true. I don't have that in the forefront of my mind. Exactly. So we have done assessment. We have used other assessment metrics in terms of their knowledge of the system of cellular respiration, not concept maps. And we have never, that I recall, plotted like basically performance on that versus performance on this at this stage.
How is the final test for this course is given? It's like a regular.
Yeah,
it's a regular university test.
Yep. We know that. So we don't have before they started using these this lesson. We do have some data that they improved in their understanding of cellular respiration, and there's a couple other lessons in there. But it's not strong because those in the lecture, so this is done in the lab, they're not really fully assessed on their understanding of cellular respiration in the lab. In the lecture, it's a part of a bigger exam, so it's a small section of those questions. And we have asked that, and they have improved with using these computational lessons, but I haven't looked at like the per student, like the correlation which I think you're asking for, which is the correlation between their performance on the exams and these. And part of the reason we have shied away from that is that these are testing kind of different things. Like in the class, all they're testing is their knowledge of fermentation or ETC. They will ask them, what are the inputs of fermentation? And so those students will circle the inputs of fermentation, which I think is fundamentally different from, can you integrate fermentation into a model of cellular respiration? Right. They are very different, but I don't have the data that would answer your question, in my head at least, right now.
Joe, can you talk just briefly about the enacted pedagogy here, right? A lot of your language says, well, we should tell them more about fermentation, right? Can you explain kind of how that modeling worked in terms of the...
Right, yeah, that's a good point. So, so actually in the labs, so we are doing this work in the labs because it's been very difficult to interact with the lecture instructors and get them to change fundamentally their pedagogy. So these lessons are done in the lab in which students are walked through, so in this case with the cellular respiration, they're provided with the model. There are questions that ask them to do perturbations with that lesson, and then the questions are, so they're working in pairs, so they are interacting around, and I'll talk about that the discussion that goes around that in the last part here. But they discuss those answers, and then they basically, so it's kind of like a dry lab experience through the simulations. So we try to integrate the active learning part of it into the lessons where they're actually manipulating. So we have so I didn't actually point it out, but in the way we've set up the pedagogy is a very much a predict, explain, or observe, explain approach, where it's like predict what would happen when this is going on, observe the dynamics, explain why, and trying to lead them down the path of understanding the mechanisms behind those phenomena that they're observing. But it is hands-off because in a lot of respects because of the just the difficulties in getting it enacted into the classroom, into the laboratory, and having good fidelity of the graduate TAs discussions of it. So we've tried to take it out of their hands, and that means that the allowance is that you're not going to do some of the things that you would prefer to do with this.
I was curious whether the perturbations that they happen to have experience with and that, right, were more focused on the glycolysis CAC kind of level and not fermentation, right? Was there?
Yeah. So just to give you an idea, like the third, so there's 3 investigations where, and the third one is they perturb the electron transport chain, so they actually put in an inhibitor in there. And the question is related to what's the impact on glycolysis. So they have to basically think about what is happening out of the electron transport chain that would impact whether glycolysis is actually taking place. And so you're kind of tracing through the model, but it's not just a direct, like, no, you don't have this. It's like, well, things are stopping. So you get into this kind of pools and flux discussion about whether this is, if you stop the bathtub from filling or if you're actually, increasing the drain size, like what's going on. So it is equally distributed among the processes, but they kind of try to go between the processes rather than just like focusing on what fermentation is or what glycolysis is, because they get that in the lab, in the lecture. They get a heavy dose. They would get a heavy dose of, like, here's what glycolysis does, and they go through all the really nitty-gritty of like transformations and things. And we don't feel like that's actually, honestly, I don't think that's very useful for the students. And so we're trying to think about it more from a modeling perspective about, let's think about the system and how it functions rather than going into the nitty-gritty of how the transformations of some of those molecules during fermentation. All right, okay. So the second one is about this, we're gonna look at how simulating actually changes or is reflected in different correlations with neurobiology. So we wanted to ask this specific question, so what are the behavioral, in this case, we're talking really about performance on this very specific task, and it's specific because of the limitations of using an MRI machine, and the neural differences that students who are simulating versus reading about this system. So let me tell you about the so this is where we're going to tie back into, we're now moving into looking at these computational models. So these computational models are like that photosynthesis lesson that I showed earlier, where they're visualizations. Those components are mathematically related to each other, and in this case through some Boolean logic rather than kind of calculus relationships. But that's not really that important for most of the students. So it allows for these quantitative simulations to take place or experiments to take place. And so you can now experimentally manipulate a variable, and since it's mathematically related, you can get an output that feeds back into their understanding of the mental model. So we say that this helps them revise both the presence of certain relationships, but also the quality of those relationships, because certain things are more important than others because of the mathematical underpinnings of that model. And so the work on neurobiology, especially related to reasoning in education, is extremely limited. So there was a review out earlier this year that had 12 studies that have really, and that's really by about 3 or 4 different groups, and ones that have tried to look at things like this or tried to understand what parts of the brain are being activated by certain processes. So one is, in both of these cases, they were comparing people who are reading about something versus those who are actually doing an action. And the first one, they're holding a spinning wheel versus one who are observing somebody holding a spinning wheel or reading about what that inertia in that case. And so they find that they activate sensory motor parts of the brain, which, as you would kind of think about, right? You're holding a spinning wheel. And then the other one is more focused on this hypothesis generation. So just reading new hypotheses or even generating new hypotheses but not writing them down activates different pathways than actually that process of trying to come up with a new hypothesis. And one of the things that you'll see throughout this is that the difficulty is the parts of our, so I am new to cognitive neuroscience, for sure. Are there any cognitive neuroscientists here? Or neuroscientists? Okay. Yes? Oh, great. So you will definitely catch some things that I am, I apologize if I mess up some of these things, but there are not many parts of the brain that have a single function. They have lots of functions, they do different things. It's very difficult, science is messy, and so it's very difficult to pinpoint what's happening for example, we know certain parts of the brain are more responsible for hypothesis generation, but to say that every one of us is going to activate the same part of that brain when you do generate a hypothesis would be faulty. And the same thing is, going into this, I didn't really understand knowledge storage very well. We're not all storing knowledge in the same place. It's not all, so when we activate and we ask you some similar question, we're not all pulling from the same places. That information's stored in multiple places around the brain. Am I all right so far? Okay, good. So the context here is different than the cellular respiration. Here we're looking at regulation of gene expression. And so in this case, bacterial, we are looking at bacterial genes within this prokaryote, or in this case E. coli. There are some genes that get activated and some that are not activated at certain points in their life. And in particular, it has in prokaryotes, they have these things called operons. So operons are groups of genes. In this case, you can see them here, that all get turned on or off by a certain switch. And so in this case, that switch is the promoter and the operator that are upstream of those genes. And so in the presence, in this case, this is the lac operon, so in that for lactose. So in the presence or absence of lactose and glucose, that either gets turned on or turned off. Or it's either turned on or it's not active, actually. And so when it's turned on, those three genes together, they produce these three different enzymes that collectively do lactose metabolism, so that you can turn lactose into glucose and galactose, which ties back nicely into the cellular respiration. The purpose of this is to create glucose that you could use for cellular respiration when glucose is not present. So if lactose is there and glucose is not there, then you produce glucose from this process so you can do cellular respiration. Okay, so that's the basics of that's probably all you need to know about the lac operon if you weren't familiar with that. And so the experimental setup is that I'll start with the simulate group here. So they read about gene expression, then they completed this table of regulators in the system just to get them familiar with the system, like what activates what gets turned off by what, because it's a little, admittedly, I'm not a cellular molecular person, but it seems like it's a little bit complicated for the first time you're going through it. So we asked them to complete this table of positive and negative regulators. This is kind of to get everyone on the baseline understanding. And then they went through the lessons. So they had to do the predict, observe, explain with a few investigations about whether lactose or glucose is present and things like that. And then, so the read group, what we did is we took that exact same protocol, and then in each of the cases where they had to make a prediction, an observation, or an explanation, we would take the output, paste it in there, and give an expert explanation. So basically, they were just reading the results. I don't actually think this is that dissimilar from what a textbook would kind of be doing, basically. It's in a little finer detail than you would see for many textbooks, but you're basically providing the analysis for the student. And then, and we'll talk about this in more detail, then so they did this all outside the fMRI, just in a room. And then they go in and they get placed into an MRI machine. So the MRI machine, what that's doing, and then they answered some questions, and I'll talk about those questions that are related to these, to systems thinking and to this, this lesson. So in the MRI machine, what's happening, just to give you some idea, so your neurons are cells, they need energy, and they also need oxygen. And so they, when they oxygenate, so they activate, they need oxygen to activate, and so that you have this these peaks where they go and they get active, and then they kind of come back down and they reset. So that's really important because what the fMRI or the MRI is doing is looking for these peaks where it's active. And parts of the brain, so it's looking, so these are just slices of images, and they're looking in these little parts of the brain and saying, what's the oxygen level? Is it rising or is it stable? And so you're comparing, when you look over this protocol, you're looking for peaks and valleys when you ask a question, what parts of the brain are being activated during that questioning. Am I still okay? Okay, is anyone else confused? Okay, so what we had was two sets of questions. We had ones that were really specific to the lac operon. So these ones we call as the system-specific questions. And then we had ones that were general, and in which case we had these questions that just had A, B, C's, and D's. So basically a very abstract system. But we've tried to make sure that we have the same, or not the same, but similar arrows in terms of excitatory or inhibitory arrows in there. So it is definitely asking students to transfer knowledge to an abstract system. That was the purpose of that. While the purpose of the system-specific was not all-inclusive to understand their understanding of the lac operon, but to have them thinking about what they learned a little bit and trying to apply it in this new, because how many people have sat in an MRI machine? Yeah, they're a little bit, it's not, it's like you have a wall like right here. It's a little bit discombobulating. And so it can be very difficult, and we understand that. But we want them to understand the ideas of the lac operon. So this is not an assessment of like learning. We're not trying to say they learned a lot or they didn't learn a lot. This is trying to understand what parts of the brain are being excited when they do that. And then, like I said, we would show them the question, but then it would become active, and that's when we would actually scan the brain, and that would say, what's happening, what parts of the brain are very excited when you answer that question. And then we go to baseline, so basically try to let it reset. And we would ask them some control trial, which was basically like some, you can see it's just missing all the words, and the questions would just be kind of oddball, and basically we're trying to get some very controlled setting about where which parts of the brain would light up anyways when faced with some of these questions, and then go back to some baseline. So we would repeat that for the system specific and then the system general.
So is that all occurring while they're in the
Yeah,
machine?
multiple times.
So are they seeing it like right?
So it's actually being projected on, so the thing about an MRI is it's an extremely strong magnet. So you have very limited, so the other thing about this is that they only have two options. That's part of the problem, is that you only have these little handheld things where you can do A or B, basically, you're active or inactive. And it's being projected on a screen so it's basically like this, and it would be projected into basically a screen in there that they're seeing. Yeah. So it's a little bit farther away in these machines, but the screen is basically right there. Yeah.
Doesn't that affect how they're going to respond?
Why is that? Which part?
I'm freaked out. I mean, I'm freaked out when I'm trying to anyways, let alone trying to figure this out.
Yeah. So the selection process is.
I mean, they're
yep.
really, and then the noises there.
Yeah. If you haven't heard the noises there, so the selection process definitely goes through all of that. There is a practice fMRI where they can slide in and see what it feels like to be in such a close confined space. There are a lot of limitations. You can't take people who have any metal piercings, even like tattoos, some of the older tattoos, they'll heat up. And so there's a lot of selection criteria that are necessary to get people that can do this. But you pay them a lot.
But it just seems like it seems like the probe might be influencing what you're trying to figure out.
I can't answer this generally because I don't know, but I mean, Carrie was okay with it and saying that it would probably be fine. I don't know. They've been doing it for years and years. I don't know. So.
But that's also the reason you have the controls in the baseline.
Yeah, I mean, within there you have like, okay, you get to hear like this. It's like 20 minutes of it's nauseating for me. I mean, they have earplugs in and all sorts of stuff, but it's nauseating just watching it happen. All right, so the results are that behaviorally, so we were looking at their performance, the likelihood that they would answer the correct answers. Interestingly, so we didn't find any difference between the read and simulate groups. So there was in both the system specific and the system general. And you'll notice that the system general actually performed no different than chance on those questions. And I'll come back to that in a little more detail. But the big finding is that there's no difference between the read and simulate group in terms of their behavioral responses. However, when you look at the neurobiology, that's where it starts to get very interesting. That when we look at system specific, so again, don't worry about the names because I couldn't tell you a lot of these names. This is why I work with Carrie and you collaborate. But there's these increased activities in certain regions that have been over time associated with this hypothesis generation and causal reasoning. So there are parts of the brain that do tend to be more active during these types of tasks. And then for the system general, again, there's increased activity in some of these regions, but these ones are associated with the sensory motor regions. And as I said, the whole idea with the system general was to try to get students to transfer these ideas. And one of the things we found was that they are definitely not using analogical reasoning during these transfer tasks. And so across the board. So there is no difference between reading and simulating. And across the board, they're just not using analogical reasoning, which comes back to, well, I'll come back to the conclusions from that. The other part that's really interesting is that, this is showing that there is neurobiological differences between reading and simulating, especially over here. And then in some cases, the simulating are doing more of this sensory motor work. But when we look at individual differences, so these are where we went in and we asked, just for the questions that you answered correctly. So we're just going to ask you, we're not going to look at the ones you got wrong, but when they make, and this is across both groups, so reading and simulating, if you got a question correct, what parts of the brain were being activated when you got those correct answers? And it is definitely consistent with the idea that certain parts of the brain that can determine plausibility, and then also with the ability to detect errors, to detect and inhibit errors. But there's a lot of individual variation here. So certain students are getting the right answers, but they're activating parts of the brain differentially. So some of them are activating it a lot and getting the correct answer. Some are activating it less and getting the correct answer. And that was, so Carrie focuses on individual variation. And so that's a really intriguing finding that you would have so much variation in terms of getting the right answer across these groups. So, so some of the conclusions here are that they are, even though we're not seeing any behavioral differences, there are definitely different brain regions that are being activated in those processes. So simulating does something different to your brain than reading. And I know that that sounds really trite, right? Like, of course we know that. But we actually don't know that. We think that. And so what we're trying to do is know that a little bit more. And so we don't, I mean, this is an initial step towards understanding this better. But for these researchers, we don't know exactly the whole amount of cognitive processes that are going on in all these parts of the brain. Like I said, most of the brain is multifunctional, and we have some better understanding about hypothesis generation taking place in certain regions and error detection taking place in certain regions, things like that. But we don't know for sure, and there's a lot of work that's still being ongoing about that. And then that there's a lot of individual differences that are contributing to this variation in performance scores. So for instructors, it's really important to know that just when you see something on paper, that there is a lot of different things that are going on in students' head that got to that. And they may, so it doesn't really explain all the activity that went on in their head. And some of these activities, so we would make the case that some of these, like simulating, is actually preparing you to do better other things than reading. So even though behaviorally they're going to perform around the same, they may not, in this very short task, right? Remember, we have just a task, and then we're going in right away and asking them about that task. So there is no long-term learning here that we're trying to get at. But it's definitely causing different parts of the brain to be activated in that very short amount of time. All right, so should I pause, or do you want to come back to this at the end too? Oh, you have a question? Yes?
And during one part of this, you said that something showed that they weren't using analogical reasoning between the two things, and I didn't quite catch the rationale for that statement.
So there are parts of the brain that are definitely, and I couldn't tell you which ones, but are more, that come up frequently with analogical reasoning. So transfer tasks where you would ask, how's the solar system like a cell or like an atom? And so students who answer that question tend to light up certain parts of the brain. And we are definitely not seeing that.
So do you actually ask the students to do the thing?
Right, so this is, so here's the, one of the major limitations. So there's a couple things. Yes, there's asking that question, which we didn't think, we thought we wouldn't, not that we wouldn't need to, but that we're just trying to see if that part of the brain is even going. Part of it is that the limitations of the MRI machine, which is a couple things. One, like you only have 2 answers. That's not very reflective of a lot of things, but that's the limitation. You can't have them typing, you can't have them coding, and you can't have them speaking. Now, you can correct for it. Like when someone's speaking, you can wait till they're done speaking and then scan it, but there's enough movement that you can't actually do it while they're speaking. You can't have them coding because you can't have all that metal in there. I have to say, like, we all go to these training things about whatever, and the fMRI training videos are scary. Like, honestly, I left and I'm like, I'm never working with it. I mean, like the metal that'll just fly across the room, the chairs that they show flying into these things, and it scares you. Like, I am not going to have metal within 100 yards of this thing. You're like pens, or like, did I forget some metal somewhere? Because it's so strong, but and it's really helpful, but it also has these detractions. Like, you have to be perfectly still. You have to, I mean, they're not strapped into these chairs, but like their arms are down at their sides, attached to these buttons. It's like A or B, and that's because we can't have anything moving up here. And so even though people think, well, you can kind of not move, but even blinking and stuff it can be distracting and affect their ability to process those images.
I was even wondering if afterwards.
Yeah, so I think that's the conclusion, which is like, can we do it afterwards? And I think that's what we need to do. And you'll see, and actually one of the conclusions for the 3rd section is, like, can we do some of this with the building of the models? And it's really hard because you can't have them do it in the things in the machine. And if you do it afterwards, like you just have to consider different things, like what's going on in the brain before, during, and after the scan. I think, as Joe is suggesting, is the noise affecting what they're actually doing in the machine has to be really something that's considered. And I'm sure other people have considered it. I don't know the evidence for one way or another. So I want to just talk about the 3rd one, and then we can definitely have time. We'll just open it up for more questions for sure. In the 3rd section, I'm going to talk about this. What we wanted to do was have students build these computational models. So they were going to build them from scratch. And we know that this is, in terms of time, this takes a lot more time than just being provided with a model and simulating it. And we recognize that. And so there are going to be limitations that you will probably pick up and say, well, that's time on task is going to be different here. And we completely recognize that because, our response is, building models takes time. Scientists take time to build models, and that's the authentic practice that they do. So we can't have a completely It's not going to be completely representative, but that's one of the limitations that we're going to acknowledge. So in this case, so I call it investigate here, but this is basically on the left side. That's the same as what you talked when I talked about the simulate. So we're using the lack operon again. So we had the group that was simulating or investigating, and then so again they had this reading the gene expression like background material. In this case, we had a pre and a post concept map or conceptual model that they build. So we're going to talk about that in a little more detail because that's one of the things we were looking at. And then they have this predict, observe, explain thing that I talked about earlier. Now the difference is that the build group, after they built a conceptual model, just drew it out, then they had to construct that in the system. So they had to go into and it was basically pretty much a blank slate in the cell collective, and it said, add your nodes, add your edges, and simulate. And importantly is this second stage here, which is that they needed to validate their model. There are only 4 conditions here. You have lactose and glucose, and they're either present or absent. And so in each of those cases, you can tell whether the lac operon is going to be on or off. And so if your model is working correctly, for each of those conditions, it should work. I mean, it's pretty straightforward in terms of the validation. Now, the tricky part is, like, what does that mean about the model? Like, now I need to take that information and revise my computational model. So that's so here they build this model as they came in, then they translate it into this network model, and then they have to simulate it. And here's the conditions: glucose, lactose, absence or present, and then whether they would expect that the lac operon would be on or off. And then they were expected to go back and revise that continuously, or continue to do that until they got the correct model. Now, we went back and forth, I mean, for days and weeks, it seems like, whether we should actually ever give them the correct model, or whether they should finally get to the right model before they could move on. And we ended up saying, well, we can't really hold students there for that long. So we do have a point where, and some students get to this where they just say, oh, well, hey, if I just push next, it just gives me the model and then I'm done and I can keep going. So, so some students shortcut this, and some students, interestingly, keep going, and it's great. They keep working on it until they get the correct one. And some will write things in here and then say it worked, and we know it didn't work. So that revision part is something we're still trying to figure out how to improve what students are actually doing that. But they are asked to revise their model if it's not functioning. So there is a fair amount of self-report. No, I mean, about what's that process. But what I'm going to talk about is the discussions that were going on while students were doing these. So I'm really interested in the cognitive processes, that shared cognitive process between as you're building these models and as you're simulating those models. So students, okay, so I'll come back to the discussion here in a second. So behaviorally, they're very similar, pre to post. The building versus investigating, if you look at the number of structures, the number of relationships, the quality of those relationships, there's almost no differences between those. But just like with the neurobiology, you should be asking yourself, but there's got to be more to this, otherwise this would be the end of my talk. And so we really want to know what is going on in the brain, or what's going on cognitively between these two groups, because it's a fair amount of different work that you're doing in terms of building a model. And we expected that there would be some differences. We noticed that if we look across all those groups, they're all adding structures, they're all adding relationships, so there's more of them moving in this direction and upwards, so they're adding that's how they're changing their models. So people are definitely involved. Their mental models from pre to post are definitely changing and not predictable, but definitely moving towards adding more structures and more relationships. And actually the quality improves as well for many of them. So that trajectory is helpful. But the question is, just like with the other one, is there a difference between the read and the simulate? And here the question is there a difference between just simulating and actually going through that whole process? Is it worthwhile to go through that whole process of building a computational model? And I would say that it is, with some caveats here. And so we use this framework on cognitive processes during models that Sins et al. Proposed back in 2005. And in which case they talk about more surface reasoning. So we have ones that are kind of no reasoning where they're not, so the instructor is talking versus, and we actually even lump, in some cases, the reading and paraphrasing into the no reasoning because they are often just listening, or someone will just read what's being in front of them. And I don't think there can be reasoning that's going on, but it's very, very surface level. And so we're really mainly focused on the four to the right. So the analyze, inductive reasoning, explaining, and evaluating. And the gradation for us is moving from vague descriptions of these concepts, things like I don't think you can connect glucose and CAP. The specifics aren't that important, but you can see that that's quite vague. Like, okay, so they're really thinking how they're connected, but that's not really telling us a whole lot of in-depth, to moving to more specific, and then finally moving into this explanation where they're trying to actually make some connections between those and provide some definition of a clarification about what the mechanism might be. The evaluation is one step further in terms of evaluating the quality of that relationship. So because no enzymes can break it down, like are we trying to understand, or I'm sorry, I should go to the previous sentence. So the lactose breakdown is inactive. So is that always inactive? Is it partially inactive? What's that strength of that relationship relative to the amount of glucose that's coming in? So there's other ways to evaluate the quality of your model that would be one step further in this progression. It's not to say that students, I shouldn't have said progression, I'm sorry. It's not to say that students are going to proceed from analyze to evaluate. They're using these multiple times during this thing. And so we're trying to look at the frequency that they are popping up with inductive reasoning versus explanations and evaluations. And we notice, so this is just the students who are building, that you'll notice that for the most part, they spend most of their time in analysis. So they tend to say these very vague terms, vague understanding of the relationships between there. And then as you move, so these color gradations move deeper reasoning. And you'll notice in particular, this is the only time in actually this entire study when evaluate starts to come into play, is during this revision stage. Is students are actually evaluating the quality of those relationships because they have to revise that model. So the revise has this evaluation, but by the time they get back to the simulate, where they're actually now they have a correct model and they're just doing the perturbations, they do this explanation here, this gray bar, when they're prompted, but they don't tend to, they don't go back to the evaluation of the quality of the model. They just assume. Now it's assumed that this is correct, and so I'm not even gonna evaluate my perturbations or how that's impacting the model. And when we look at, so this would be comparing the build versus investigate. I'm sorry, I should have reversed that order from earlier, but the investigate students, now they do a lot of analysis, so they didn't get that opportunity to build a model. So they are doing a lot of analysis that the other students have already done. Remember, those students have already analyzed the bejesus out of all these relationships. And so these build students tend to do more of this explanation than the investigate students, but they still, you can just see a smidgen of a yellow bar there, that they do some evaluation, but it is extremely infrequent. And the investigate students never evaluate. So they never reach that evaluatory stage. So the investigate students, they do have more analysis and inductive reasoning, but that's probably because they haven't had the opportunity to do that prior to this. So if we did look at time on task, so this is the number of discussion, like utterances basically, or sentences or kind of phrases that students are having. When we looked at time, the result is actually quite similar to these. So they are investing a lot of time doing the analysis, these investigate students. But that's because they just haven't had that time going up to that. Now, like I said, the build students do invest a lot of time in that build stage. It takes about an extra 45 minutes for them. So this becomes about a 2-hour endeavor versus an hour and 15 minutes kind of endeavor. And that might also be part of the reason that we see this kind of tailing off here, is that students do get burned out. And that building actually takes a lot of energy. And so the quality of the students' actual discussions later and some of their answers tend to decline. So that's the downside of that. So, so behaviorally, again, there was very little difference between investigating and building. Again, the short interventions don't necessarily result in like big changes. I mean, I don't think that's completely novel. It's just a reiteration of that there's more evidence that short interventions aren't going to be the trick, but that what's happening is going on in the brain or cognitively, that they're doing different things, that they're actually performing more model evaluation when you're building a model, and that those might have long-term effects. So you might ask, as I was kind of suggesting earlier, why not look at these neural patterns for this investigate group, or this build group? And we did start to do that. We had a pilot study, and they were showing some changes. One of the reasons that we only did the read versus simulate group is because of expense. So fMRI is extremely expensive, and because of the time on task. So like I said, it took an extra 45 minutes. So if you think about a student coming in, so they're almost spending 2 hours, and then they have another hour in an MRI machine where you're getting boom, boom, boom. You know, the students were totally wiped out. So they were, we were really hard pressed to continue that study. We need to think about how to do that differently in order to really look at the neural patterns of the building group. And again, you can't really have them build in the machine, although that's the person who's not a cognitive neuroscientist saying, yeah, let's do this. And then she goes, no, it won't happen. It can't happen. So overall, reading is really a critical process. I don't mean to put this down. It is really helpful to get that declarative knowledge. There's a foundational amount of knowledge that's necessary to kind of lay the foundation. I don't want you to think that that means that you need to have your students read everything before they can do any modeling. Actually, not that at all. But you do need some amount of information in order to organize it. And that's what, so I don't really have the conceptual modeling in here, that this building conceptual models is really critical to organizing all that declarative knowledge. And students need practice at doing that, because we just take for granted that they're going to figure out how to organize it. But there are a lot of gaps in there that we can, as instructors, can pull from the students, and they can pull for themselves. The simulating really activates these different brain regions than reading can. So it's regions that are associated with causal reasoning and hypothesis generation, and also error detection, their ability to detect errors. And then building leads to more model evaluation. So probably during this revision stage is when we're really getting different cognitive processes are taking place, because you're asking them to revise the model, their mental model, that feedback to their mental model. So with that, I'll take questions if you have questions. And I appreciate you guys taking the time. I've really enjoyed my visit, and this has been great. So thank you. Yes?
Can I ask a question about the brain regions when you contrast in an opposite way, reading versus simulation group? Because the contrast is based on the more activation and simulation group. What about the other side? Reading group, more activated in a particular
I don't have that off the top of my head. I have it in the paper on my computer somewhere. I don't know the rate. I mean, if you stick around, I can kind of show you what the results are on that side of it. I don't know which brain, and I couldn't even tell you, I couldn't say the word. I could say some random brain regions, but I wouldn't be right. So yeah, but if you stick around, I can show you what information we have about that contrast, I think.
Thank you.
Yes?
Just to have the clarification question, is the building means building conceptual model and building the simulation model the same thing?
That's a very good question. No, I'm sorry about that jargon. So the building here is I'm specifically talking about constructing a computational model. Yes, I would, but you point out a very good error there, which is that, drawing, we'll say drawing a conceptual model, I would kind of put somewhere, that is very helpful in terms of organizing information. But I'm talking here about building in terms of actually constructing a computational model that you then revise and work with.
Now this makes sense.
Yeah, thank you for asking that. Yes?
You showed a scatter plot of connections and quality,
Yeah.
right? And you showed that all the students kind of move towards the
Well, most.
quality.
Yeah.
What happens if I color code that based on the two groups that you had in the experiment? Would I get a uniform distribution?
Yep. Yep. Actually, yeah, that was one of the. So I did that analysis, and there is no change. There's no. It would look a lot like that blue and green pre-post kind of thing. That they all. It's kind of mixed around. Some are really improving that are coming from the simulate. Some of them are build, but there's no uniformity there.
But maybe those are like different clusters of students. You can consider.
Yeah, I mean.
Use cluster analysis and say, well, this group, regardless of how you put them, either reading or simulating and analyzing, they will do well. This group, maybe there's some underlying feature to those clusters.
Yes, you're right. I did not do a cluster analysis. I have looked at it. It is pretty uniformly distributed, but I haven't done the analysis to back that up. No, that's a really good point. Yeah.
So in a follow-up question to that sort of graph, so the students are adding more stuff, more connections, more boxes, and they're of higher quality, which is kind of, I don't know, maybe somewhat surprising if we think about experts who might actually prune their models to just a few ideas. And so I guess, is the stuff that they're adding actually relevant to the function of their model, or are they just adding more stuff they heard about in class because they think it should be more complex? Does that make sense? Like, can you evaluate that part?
Yeah. So there is. So this would be. So that data is relative to pre-post around the lesson. So they wouldn't be adding any more necessarily that they learned in class, but they could in the lesson. Yes. And I think that's part of the process. It is the process of learning, right? You just read about something and worked with it for an hour and a half or whatever. You're going to add more things to it. But that the quality increases is interesting to us because you're not just adding it peripherally. So now you're actually starting to integrate it because those relationships are of higher quality. And I think, so I think you're right about the pruning. But what I think is happening is that we're at the early stages before you prune. So that you would start to, I think if you repeated this over time, and this is what we were kind of showing in a much earlier paper, that if you did this repeatedly through the semester, that you start with the same types of models, that you would start to prune those or improve the other quality of some of them or get rid of certain relationships. And so that's why, one of the things, or on this slide here, so I talk about them relating components linearly. There's actually not an analogous thing here because I think that we have this idea that experts have this beautifully interconnected model. And it's more interconnected than a linear model, but it's not like a spider web. So we actually use this, this web-like complexity analysis. And I think it's not really that all of the things are interconnected with everything else because I don't think the systems are exactly like that. We do want them to show more connectivity, but there's a limit there too, right? Like biologically, we can't just put arrows everywhere. And I think that's what the pruning happens over time, which is I don't really need this arrow, or this arrow is weak here. And so we should probably either invest more effort in understanding what that arrow should be or prune it. And I think that does happen over time. But I think that this intervention is not getting at the pruning because that is something you need knowledge consolidation over time. Like, you need more information than a 2-hour lesson is going to get you in terms of pruning some of those things. I think that's my take on that. Other questions? Yes.
So I'm thinking about your second fMRI study and how you did the domain specific and then gave them the domain general question.
Yeah.
I was wondering if you guys have thought about why you chose that order and why you didn't do the general first and then the specific.
I don't recall the decision why we did it in that order. I would say that, so part of the reason we never did that complementary study is just numbers, right? So, but I think it raises a good point about what you learn from the first one that you take to the second one, which would be
vice versa. Because
yep.
I'm thinking about how you learn it in class. Like typically you get this general, like, hey guys, here's where we're going, and then you go into the specifics. And this seemed almost backwards to how we might think about it in class. Yeah.
Honestly, I hadn't given it like too much thought about that. I don't remember exactly why we made that. And it's a good point because there's definitely, yeah, I don't know. I honestly don't know exactly what would have happened, how it would have affected their performance on those. I think that those questions, as you saw, like they performed at chance, and I think that they got complicated very quickly in terms of, even though it didn't seem exceptionally complicated, I think and I think going back to the time thing, after a while, I think there's tiredness that is creeping in there. That's like, I don't know. It's just I'll just push A, yeah. And that's something that if you'd reversed the order would have accounted for that. I think more systematically. Yeah.
So I'm just going to talk a little bit about the nature of models. See what you think about this.
Okay.
So for me, if I'm going to draw a paper pencil model, or if I have a computer-based tool, right? I'm going to use either my paper pencil model, paper pencil, or my online tool to basically construct a model so that I can either explain something or predict something.
Right.
Right? Both of them are doing this. There's some advantages to the computational tool, but there's also some advantages to the paper pencil thing, right? But both of them have the same outcome. Right? Outcome, well, I'm trying to explain something.
Okay.
I'm trying to construct a model that it can explain it to, right? And the online tool might have some advantages because it can generate some data.
Sure. Yeah.
Right? Where my paper pencil one can't, but I can certainly check to see if my relationships, the set of relationships I have, does it really explain my phenomena or not? Right? I don't have that check on generating data, but I can say, is this working, right?
Can you though? I mean, like if you put it on paper, I think that you...
If I take out a relationship, it no longer works.
Okay, if you take out a relationship, but you don't know about the quality of the, like, so just putting arrows on there doesn't tell me you understand how all those things really go together.
I could say, as this thing increases, this one's going to decrease by such and such amount. I mean, I could do that when I'm drawing a paper pencil model.
But it's hard to hold that much information in your head.
That's why it's paper pencil. You're writing it out on your little...
Sure, okay. But sure.
So I'm just wondering so for me, it seems like you can take part in a lot of evaluation building a paper pencil model.
Yeah.
And so it seems like it would stimulate my same parts of my brain as if I'm doing a computational based model.
I think that yes, I think that you're generally true. I think, so I don't have anything to draw on, but I think where it breaks down a little bit is where that computational models have a really significant advantage is when you have things where you have like multiple relationships that are affecting things. Sure. Because there is a capacity for our brain in terms of its ability to process those kinds of interactions.
I think there's some wonderful advantages to a really good online tool, right?
Sure, yeah.
Generation of data. But it just seems like, to me, a paper pencil model, which you want to call a conceptual model,
sure,
can also allow you to do some of that same kind of thinking.
Yes, yes, I totally agree with you. And in fact, one of my pushbacks with Tom has been that he wants to have everything be contained within that software, right? That environment there. And I have long said that, for example, the LAC opera in the building, where it's like draw on a piece of paper what the conceptual model is an essential first step to being able to do that in the computer. Because yes, you could just go to the blank slate on a computer and start putting boxes and arrows and see if it works. And we know that doesn't work. You need to sit there and actually conceptualize what does this look like in order to now translate it into that. Because there's a translation that has to take place. And so I do feel, I strongly feel that drawing a conceptual model gets you a lot of ways towards being able to do it computationally. And I think that experts for sure do this. It's just not explicit to people necessarily because we don't ask them to. But they're visualizing what they're putting down on paper without putting it on paper, I think, oftentimes before they're translating it. But I think that's not clear to students that they've basically worked out everything on paper. It's just that their scratch pad is in their head. And so it is I do agree that you can do almost everything on a piece of paper, and that the computational model is just an additional, it helps you make a little bit more step towards what I would think of as an expert thing, which is holding a lot of things at once that you can't really do on a piece of paper effectively, like to say, glucose and lactose are coming in at multiple different rates and all these other things that a computational model has no problem working with. That stuff becomes very difficult on a piece of paper. But I completely agree that what you put on paper is basically translated into a computational model and is really effective. Because I do think it's more than just organizing the knowledge, even though I talk about that here.
Right, that's what I was trying to get at. I guess I was disagreeing with you that that a conceptual model is just organization, right? To me, building a paper pencil model, and that's kind of why I changed terms on you, is because you can still do lots of explanations and predicting. Computational models or online tools are great because it can generate data that you can then compare it to actual data, but you can't with your paper pencil.
That's true.
But you can still go through some of your same thinking processes. You guys in your class did some beautiful work with paper pencil models. Right?
Yeah,
it's
sure.
Not like they're not.
Oh yeah, I totally agree. Yeah, so I'm sorry if I didn't mean to suggest that. For sure, I did not mean to suggest that the conceptual models are something that we shouldn't be doing more often and doing more with. Yes, I did not mean to come across with that for sure. Yeah.
Yeah, the way I see it is like when you're drawing with pencil and paper, you can propose hypotheses or propose predictions, but you can't test them.
No, that's correct.
Yeah. So
That's the limitation.
Yes, there you go, right.
It's really nice for that, right? But it's not like you can't do some really good thinking with.
No,
yes.
It's against the precursor. Yeah.
We should probably let folks go. I really appreciate that everyone stayed for this is a really good discussion, and I know that Joe is actively putting together ideas to keep moving this work forward, so I really appreciate the feedback. And on behalf of Create,
thank you.
Thanks.
Excellent.
For your time.
Great, thank you.
If anyone does want to follow up and have additional conversation, Joe is able to hang out at least for a little while, right?
Yeah, I'm not sure exactly what's
where your
next, so
thing is.
yeah.